Okay, my name is Sydney. I am a rising senior from the University of Florida, majoring in chemical engineering. And this summer, I worked at the University of North Carolina at Chapel Hill in the Department of Pediatric Pulmonology in the Haygood Lab. And my summer project is titled Lung Epithelial Cells and Fibroblasts for In Vitro Models. So there's kind of two parts to my project. So starting with the first part here, for the background and design, we are focusing on fibroblast cell culture. So a very common lung disease is idiopathic pulmonary fibrosis, and it is a very aggressive lung disease. After diagnosis, life expectancy is only about 3 to 5 years. So we are working on a drug screening model to test Thy1, which is a drug that was found a while ago. So we started off with human fibroblast lung cells. We plated them and treated them with TGF-beta, which is a cell growth factor, and this helps so that the cells turn into more of their fibrotic state that is commonly seen in pulmonary fibrosis. After that, we treated with Thy1, both Thy1 FC and 833 Thy1, which are just two different derivatives of Thy1. And then we extracted RNA and ran qPCR to quantitatively validate gene expression levels. So first off, we started with the validation of TGF-beta 1. So we had a bunch of cells in store in the liquid nitrogen, and a lot of them are older than I was, so we had to make sure that they could grow up properly and properly respond to TGF-beta before we used them for the drug screening with the Thy1. So if you look at the images on the right, you can see that there is a difference between the control and the TGF-beta treated cells. So with the TGF-beta treated cells, there is portions that are marked in yellow, and that's to highlight the spaces and the gaps that were found in the plates after treating the cells. So we can see with the control, the cells are about 90 to 100% confluent. They are showing long spindle-like structures. And then with the TGF-beta treatment, it seems as though the cells are less confluent, approximately 60 to 80%. And that is because the TGF-beta, when the fibroblast cells start to grow, they move into more of the myofibroblast type, which is a more circular structure. And we can see that at the bottom with the SMAD4. So this is a cell marker found in the cytoplasm of cells. So you can see it's a little bit more spread out on the left with the control fibroblasts, and on the right it is more centered in kind of more circular form. So this just shows that the cells are moving from their spindle-like structures into more circular structures. And this was also validated with qPCR, where we have our ACTA2 and collagen 1A1 expressions, which both went up with the TGF-beta treated cells, and that's exactly what we wanted to see. So then we moved into our drug screening, and again, we measured our outcomes using gene expression with ACTA2 and collagen 1A1. And our purpose with this was to see these gene expression levels go up from the control to the TGF-beta treated cells and then back down again when treated with Thy1. We treated them at a few different nanomolar values. And our results from these experiments suggest that Thy1 can serve as a strong candidate for continued drug screening and future clinical translation to reduce the effects of pulmonary fibrosis. So then the second part of my project was with AT2 organoid cell culture. So AT2 cells are alveolar type 2 cells. These are in the most far out parts of the lungs where the gas exchange actually occurs. And with our design of experiments, we started by digesting the human lung specimens into either single cells or clumps of cells. And we wanted to isolate specifically the AT2 cells, and we did so using magnetically activated cell separation. And then we created organoids and harvested those in a Matrigel cell culture. So, after starting with the enzymatic digestion, we wanted to isolate specifically the HT2 280 positive AT2 cells. And we can see that this was validated by both flow cytometry and by immunofluorescence staining, where we see the HT2 280 as a cell surface marker. And you can see there are yellow areas on the outside of those cells, confirming that we did have the correct HT2 plus AT2 cells. From there, we created the organoids, and these are shown on the left in just a phase contrast microscope showing the clumps of the cells together. And this was validated by pro-SPC staining, which again confirms that our organoids were the proper AT2 organoids. So, being able to successfully culture and characterize our AT2 organoids enhances our lung culture methodology and supports their application in future drug screening. So I would like to thank my mentor, Dr. Koji, to my Dr. Haygood, whose lab I worked in, Dr. Randall, who was the other PI that I worked under, and the tissue core staff at the Marsico Lung Institute. And I would also like to thank the HuBMAP program and the NIH for funding this opportunity. And that's all.